What a fishing rod we have here, isn't it? And we usually think of fishing as purely sport. But at the Hanford Atomic Products operation in Richland, Washington, on the bank of the Columbia, we do a type of fishing that Isaac Walton would never have dreamed of. This work is done by the Aquatic Biology operation, which is headed up by Dr. Richard F. Foster. We have Dr. Foster with us now, and I'd like to introduce him. He's manager of the Atomic Products, or the Aquatic Biology operation. How do you do, Dr. Foster?
Hello, Frank.
I'd like to ask you a blunt and point-blank question to begin with. If it's unfair, just say so. What is the work of your Aquatic Biology operation?
Well, this is really quite simple, Frank. Aquatic biology has to do with the study of life in the water. And here at Hanford, the largest body of water which we have is the Columbia River. And so our work is mainly with the life in the Columbia. Now, I expect your next question may be as to why should we at Hanford study life in the Columbia River? The answer to this is also quite simple. We have a unique kind of effluent from the Hanford reactors. Water from the Columbia River first goes through the reactors in order to cool them. It is then returned to the Columbia River. Some of the dissolved minerals which are present in Columbia River water become mildly radioactive. And we would like to know when this material gets into the Columbia River whether any of these mildly radioactive materials are picked up by the aquatic life, and if so, where it becomes deposited.
You've given us some indication of what your work is about, Dr. Foster. I know that much of it must be accomplished on the river. Would you tell us what kinds of aquatic life you encounter in your work on the river?
Yes, Frank. The kinds of things which you find in the river are much like those which you find on dry land. We have the green plants represented in the river by algae. Some of this algae is present as tiny floating cells present in the river water. Other types of algae grow on the rocks. We have many different kinds of organisms, such as the snails and insect larvae, which eat on the algae on the rocks. And of course, the fish may feed on these kinds of life which grow on the rocks. Other kinds of fish feed on other kinds of fish. So our problem really is that to study all kinds of life in the Columbia River.
Well, I happen to know that as you work on the Columbia River, you gather specimens, and you brought along some preserved specimens, which are rather unusual. Would you show them to us now?
We brought along a few, perhaps some of the more unusual kinds. The plankton algae are too small to show very effectively. We do have clams present in the Columbia River. Clams were once used in the Columbia by Indians. We also have crayfish in the Columbia River. Crayfish which grow to a large size, but for some reason or other they're not a very popular food for people. We have two different kinds of fish. The small bullhead, which lays in wait at the bottom of the river to capture other fish which may swim by. And of course, a couple of very small specimens of the sturgeon, which is the largest fish in the Columbia. I'm sure many of our viewers have already seen pictures of the mighty salmon, which are very common and very valuable to the Columbia River system.
Well, Dr. Foster, it's quite easy for any of us to understand how you gather samples of this size, but as I understand it, the plankton are very small. They're almost microscopic. How do you gather those out of the river?
We have to strain these out with a net which has an extremely fine mesh. These nets are made out of silk bolting cloth. Their mesh is so fine that they strain the majority of the organisms out of the water, but some of the organisms are so small that they even go through the mesh of these kinds of nets.
Mm-hmm. Now, after you do gather these plankton samples, I imagine you take them back to the laboratory and do something with them.
This is right. We're interested in the radioactive materials which are present in the plankton and also in the other forms such as the fish. When we get these materials back to the laboratory, we reduce them to an ash, sort of a cremation process. This is so that we can put them in a counter and count the amount of radioactivity very effectively. We put these on a small plate, which is about the size of a quarter, and these plates then are placed under a Geiger tube. We can rather accurately determine the amount of radioactive materials which are present.
I see. Now, in your work on the river, do you make any other tests that might be interesting and are significant in your work?
We're, of course, interested in the water because, after all, what affects the water affects the organisms. And so we take samples of the water. Sampling the water is rather simple at the surface, but when we want to sample at depths, this requires special equipment. We have here a bottle which is used for sampling water at any desired depth. We send down a little metal weight which trips the ends, and we can bring the water back up to the surface and know the situation at any particular depth. We're also interested in current, how fast the water goes by the organisms, because they are affected by this. Certain ones like fast water, others like slow water. We can measure the current with a current meter, which works on the same principle as the anemometer, which is used by the weatherman. These are only a few of the various kinds of things which we do.
Certainly is very interesting, and I've enjoyed, and I know our viewers have enjoyed this brief resume of your work on the river. I know you didn't tell us all the work. Now, I know that you must be doing some work in your laboratory since you are a scientist and are working with scientists. Would you tell us something about your laboratory work, Dr. Foster?
Well, the work in the field would not mean very much without correlating this with the work which we do in the lab. This is because the amount of radioactive materials which are present in Columbia River organisms are so low, we don't really expect to see any effects. In the laboratory, we can use mixtures of the effluent which greatly exceed those which would ever be present in the Columbia River. And we can use fish such as this, these little white fish, to tell us how strong a mixture of effluent could be tolerated by fish in the Columbia River. We can also use these kinds of fish to trace the radioactive materials through the various tissues. By feeding these fish radioactive food, we can find out then how much of the material is picked up and where it is deposited. So our laboratory program works in conjunction with the field survey program.
I see, Dr. Foster, I'm going to be awfully unfair now, but in 15 seconds, could you tell us the significance of your work?
The work in the Columbia River is this, the results are this. We do find that these fish pick up very small amounts of radioactive materials. The amount which they pick up is not at all hazardous. So we know that from this angle, the operation of the plant is quite safe. Similar programs are also carried on by other components for the atmosphere, for the soil, for the drinking water. This are part of the safety program which ensures that the environs of the Hanford works are maintained in a very safe manner.
Thank you very much, Dr. Foster. We enjoyed having you on Hanford Science Forum and telling us of your fascinating and I'm sure significant work at Hanford.
Glad to have been here.
And now I'd like to introduce our science student of the week, who is Mr. Burke. What is your first name?
Doyle.
Doyle Burke. Doyle is a senior at Columbia High School, and last year he participated in the Mid-Columbia Science Fair and won a prize with this critter here. Doyle, what is this critter here, and what prize did you win?
Well, this is a dog skeleton, and it was my entry into the Mid-Columbia Science Fair, where I placed first in zoology and also grand prize of the fair.
Well, congratulations, Doyle. Now, how did you happen to think of assembling a dog skeleton?
Well, we had some science workshops where, through the efforts of GE and the Atomic Energy Commission, I was able to be introduced to this idea, and with their help and advice.
You plan to go into science as a career, I understand. What field?
Well, I wanted to go into the applied science as a career where I could use both physics, chemistry, and biology, and I chose for that profession the application of forestry.
We have been dealing this evening with atomic energy. Are you interested in atomic energy at all?
Well, I think that anyone who is interested directly or indirectly with the application of science would have a definite view as to the use of atomic energy in its aspects.
Well, Doyle, I certainly want to congratulate you, and I'm sure anyone that sees this piece of work here would on this particular piece of work. And we also wish you every success in your scientific career, and it was a pleasure having you with us. Good luck.
Thank you.
Hanford Science Forum is an educational feature of the General Electric Company and the United States Atomic Energy Commission at Hanford.